Today's presentation will discuss xenotransplantation. First, it is important to understand what this means. Xenotransplantation is transplanting a non-human or animal tissue into a human recipient. This video will focus on the role of porcine tissue specifically in xenotransplantation. First, let's discuss what drives the need for this procedure. The demand for human organs far exceeds the supply of those that are donated. According to the FDA, currently 10 patients die each day in the United States while they are on the waiting list to receive life-saving vital organs. Porcine organs can grant extra time to these patients while they wait on the transplant list. It is also important to recognize that synthetic and mechanical materials in medicine may not be adequate. Let's discuss why porcine tissue is a great candidate. Number 1, there are a large amount of anatomical and physiological similarities. This includes similar organ size and physiological function. Number 2, it avoids many ethical questions. Since pork is already utilized as a food source worldwide, it is less ethically problematic than using a cadaver or a primate tissue source for transplantation. Number 3, easily accessible. Swine are very reproductively efficient and are prolific breeders, resulting in a large pool of potential donors. Number 4, beneficial genetic modifications. Genetic modifications make pork tissue less likely to be attacked by the recipient's immune response, therefore decreasing the rate of rejection. Number 5, lower risk of infectious disease. Though cross-species infection from a retrovirus is still a concern of the FDA, porcine tissues are less likely to cause disease than primates. Number 6, more economically efficient. Due to the large pool of availability in donors, it is more economically efficient to use something that's already mass-produced. Where do we use porcine tissue in medicine? One application is in skin grafts. Porcine skin grafts are referred to as xenografts because they are transplanted between two different species. They are used to stimulate granulation of tissue and re-epithelialization in large burns. They can also be used as a temporary dressing in burn patients because they ready the site for a human tissue graft and prevent the loss of insensible fluids and prevent infection as well. Porcine tissue is also used in abdominal reconstructions. Porcine tissue is preferred over synthetic meshes because it retains the mammalian extracellular matrix that acts as a natural scaffolding for host cells. It is also safer to use in infected fields, unlike synthetic meshes. Finally, we will talk about the application of heart valve replacement. As this diagram shows, there are two different types of replacement heart valves, both tissue and mechanical. Tissue valves are made from bovine and porcine heart valves, as well as the pericardium. These valves work very efficiently in the human heart and can last on average from 10 to 15 years before they begin degrading. They have a large advantage over mechanical valves because they are closer to a human heart's natural environment. They also do not require the patient to be on lifelong anticoagulants, which will increase your patient's risk of bleeding. Tissue valves are treated before implantation and do not require anticoagulants at all because they do not aggravate the body's immune system. Current research is being extended in many areas. One potential application is using neurological cells to treat diseases such as Parkinson's and Huntington's disease. Corneal transplants are also a viable option. Also, research is being done into extracellular matrix. These matrices will act as a natural scaffolding and support tissues within organs. They are experimentally being used in people.
There are thousands of people who owe their lives to pieces of dead pig tissue, and no, I'm not talking about the admittedly life-affirming taste of bacon. For the last 50 years, we've been replacing damaged human heart valves with pig valves. So if we can transplant heart valves from pigs, why not entire hearts, or kidneys, or livers, or lungs? How come you can walk around with a pig's heart valve inside of you, but you reject a kidney from another human? And out of all the creatures in the world, why pigs? So many questions. Transplantation of organs from one species to another is known as xenotransplantation, and it turns out to be pretty bizarre. But since there's a shortage of human organs, figuring out how to get a steady supply of animal organs and tissues that we could use is something we might want to figure out. Transplantation from human to human is tricky enough, much more difficult than matching blood types. The new organ is often rejected by the recipient because of a protein called human leukocyte antigen, or HLA. Almost every type of human cell has different kinds of these proteins on its surface, and normally they're the immune system's undercover agents, marking targets for it to attack. So if you transplant an organ from one person to another and the recipient's immune system recognizes the donor's HLA as foreign, it'll attack the transplanted tissue. This can lead to severe blood clots, failure of the new organ, and all kinds of painful symptoms that put together amount to transplant rejection. The problem with these HLAs is that they are incredibly diverse, with thousands of variants, which makes it very hard to find a donor whose chemistry matches a recipient. Some people spend years on the waiting list, others never get a match at all. But there are a few ways around the problem, and one of them is to skip humans and move to pigs. You might not find this too flattering, but you have a lot in common with a pig. Your immune system functions in a similar way, and your organs mostly do the same things. And pigs are the right size. You wouldn't want a heart valve transplant from a mouse or a beluga whale. The main thing is that a heart valve doesn't need to be alive to do its job. It's really just a piece of tough, flexible connective tissue that acts as a glorified piece of plumbing to direct blood flow. So if you blast the pig valve with a chemical called glutaraldehyde, you could preserve it while killing all of its cells in the process. Dead cells can't produce those HLA proteins, so there's much less risk of rejection. The risk is not zero, though. These heart valves can wear out over time since they're dead and unable to replace damaged cells. Also, sometimes the patient's immune system still gets nervous about having this foreign thing in the body. The dead cells still have some antigens on their surface, and patients, especially those with stronger immune systems, can react to them. The valve may eventually fail. So scientists figured out how to minimize even that small risk. They identified the main antigen that our immune systems attack in pig tissue, called galactose alpha-1,3 galactose, or alpha-gal for short. Then they genetically engineered pigs to not express alpha-gal so the human immune system can accept the pig valve much more readily. But why stop there? Why not use pigs to grow not just heart valves, but whole hearts or anything else you might want? That could solve the organ shortage. Well, scientists are working on it. Knocking out alpha-gal alone is not enough to convince a human body to accept a whole organ from a pig, but some more extensive genetic engineering could help. For example, you could remove the gene that tells the cell to produce some of its other antigens, or throw in some human genes to make the tissue seem more familiar to the human immune system. They've tested these genetically engineered pig organs in non-human primates like baboons, which only survived for a short time, but the technology is advancing quickly. The downsides? Some people aren't comfortable with the idea of genetically engineering pigs to express human genes, even if it's only to trick the human immune system. Plus, cloning enough pig cells for the technique to work is mega expensive. For now, these genetically engineered pig organs would be too costly to reach any patients who aren't like at the private jet level of wealth. There are alternatives in the works, though. One concept hinges on the idea that there is virtually no risk of rejection when an organ is made up of the patient's own cells. So say you take an organ, like a lung, from a human or animal donor and remove all of its cells except the connective tissue that gives the lung its shape. Then you replace those donor cells with stem cells from the patient. Cells love to stick to that connective tissue, so they'd move in and start dividing and differentiating into the proper kinds of cells. This too has been tested in animal models, but researchers still have a ways to go before they can build a completely functional organ. So the science of xenotransplantation still has a long road ahead of it. But eventually the day might come when we will be able to make organs to order, and it will likely be thanks to our porcine friends. Thank you for watching this SciShow Dose, brought to you by our patrons on Patreon who help make SciShow possible. If you want to help us keep making videos like this and get some